Welcome to our molecular biology presentation.
We are Ranjit Bakar
and Jeremy DeLuna.
The paper we are looking at focuses on the effects of microRNA on gene expression in the postnatally developing monkey hippocampus. Gene expression, the process by which information from a gene is used in the synthesis of a functional gene product, usually proteins, but can also be a non-protein coding genes such as rRNA, RNA, or even miRNA. Mature microRNAs are a class of naturally occurring small, non-coding RNA molecules. There are about 21 to 25 nucleotides in length and are partially complementary to one or more messenger RNA. Researchers has found that its main function is to downregulate gene expression in a variety of manners. They can silence cytoplasmic messenger RNAs, either by triggering an endonuclease cleavage by promoting translational repression, or possibly by accelerating messenger RNA decapping. One final but very important feature of miRNAs is their ability to be transferred between cells via specific transporters, thus potentially effectuating the regulation of gene expression in neighboring cells.
This paper, it is presented that by using experimental genome-wide analysis of gene expression and predictive bioinformatics analysis, it is suggested that microRNAs may contribute to the co-regulation of gene expression in different cell types. To test this, the scientists gathered monkeys of different age groups and collected brain matter to analyze the effects of microRNA on the development of the monkey hippocampus. 16 male rhesus monkeys were used for the study. 4 one-day-olds, 4 6-month-olds, 4 1-year-olds, and 4 6 to 12-year-olds. A variety of ages were used to compare the effect of microRNA downregulation from younger ages to older. These monkeys were also used in a previously published study by the same group of scientists where they identified genes that may be affected by the downregulation of microRNA, specifically CA3 and CA1. These monkeys were euthanized with an overdose and injection of sodium pentobarbital. The brain matter was rapidly extracted. 5 mm thick slices of the brain were cut and stored overnight in RNA later. RNA later is an aqueous non-toxic tissue storage reagent that rapidly permeates tissue to stabilize and protect cellular RNA. These samples of RNA underwent microarray analysis. Microarray analysis is a very common technique used to measure and quantify levels of gene expression. It works by introducing the sample to a solid surface containing DNA probes. These DNA probes are specific DNA sequences which hybridized the sample and allowed them to be quantified and detected. An advantage of microarray analysis is that it can measure multiple genes simultaneously, which is incredibly useful and time-saving. Microarray analysis involves breaking open a cell, isolating its genetic contents, identifying all the genes that are turned on in that particular cell, and generating a list of those genes. With this information, scientists use several different microRNA databases, for example, microRNA.org. Using different microRNA databases, they were able to determine the predicted targets of the known microRNA. The microRNA then underwent functional analysis using DAVID, which stands for Database for Annotation, Visualization, and Integrated Discovery, a free online bioinformatics resource that provides functional interpretation of large lists of genes derived from genomic studies.
From DAVID tests suggested that miRNAs may contribute to the regulation of gene expression during early postnatal development of the monkey hippocampus. They identified 65 miRNAs present in humans and monkeys as the primary regulators of the expression of a group of more than 1,000 genes exhibiting a downregulation of their expression. Importantly, these 65 miRNAs may contribute to the downregulation of the expression of different groups of genes, enriched astrocytes, neurons, and oligodendrocytes, thus providing a fundamental means for the co-regulation of gene expression in different cell types.
35% of the microRNAs identified as regulators of the 1000 Dan group of genes are primate-specific. These microRNAs could contribute to some of the structural and functional differences observed between primate and non-primate animals. The expression of 10 of the 65 microRNAs that are identified in this study has been detected in samples of adult human hippocampus containing different subregions. This study is also the first to reveal a significant positive correlation between the number of microRNAs targeting a group of genes and the degree of downregulation of these genes. Importantly, we evaluated this fundamental mechanism of regulation of gene expression in a normally developing system. Thus, many microRNAs might act in concert to regulate the expression of a large ensemble of genes involved in the postnatal, structural and functional maturation of the primate hippocampal formation. This study revealed that a large number of genes were downregulated in the hippocampus, specifically CA3 and CA1, of older mature monkeys, as compared to young developing monkeys. They considered how the specific decrease in the expression of certain functional groups might regulate synaptic plasticity and thus contribute to the emergence of a more selective and efficient memory process.
I believe this study gives great insight to the ever-increasing research into the effects of microRNA in the regulation of gene expression. However, it may seem controversial because of the use of animal subjects, and especially the unavoidable death of the subject. The predictive bioinformatics analysis suggested that miRNAs may play a fundamental role in the co-regulation of gene expression in different cell types that contributes to the normal postnatal development of the primate hippocampus. Furthermore, 35% of the microRNAs were specific to primates and humans, which can give further understanding of the developmental processes of the human brain. This mechanism is partially conserved across species, and may thus contribute to the similarity of basic hippocampal characteristics across mammals.